Well, good afternoon, everybody. Welcome to the 2025 Autumn Webinar Series for the Northeast Aquatic Plant Management Society. Let's go on our next slide here. So I'll give you a little bit of background about our society. Our mission is to assist in the management of aquatic vegetation, to provide for the scientific and educational advancement of members, to encourage scientific research in all facets of aquatic plant management, to promote an exchange of information among members, and to extend and develop understanding in this discipline. Our members are from academia, government agencies, private consulting firms, herbicide manufacturers, and lake associations. We have annual meetings, annual plant camps, webinars with CEUs as you're attending today, and we also offer student scholarships up to $10,000. If you'd like to learn more about the Society, I encourage you to visit www.neapms.org. So on to some little bit of housekeeping about today's webinar. So there will be a webinar set to run from 1 to 3. There will be 2 presentations with actually a block of 45 minutes, a block of 30 minutes at the end for questions and discussion with presenters. And as Emma has mentioned, please type your questions into the chat box and the moderator will read them during the Q&A session, which is me. Please note that we are able to offer CEUs for this webinar. Again, make sure you get those credits through Teams. The webinar will be recorded and is currently being recorded, and we will share a link via the NEPM's website very soon, where we'll post all the slides. Today's presentation and speakers, our first speaker is Dr. Bethany Bradley, talking about the implications of climate change for invasive species, and then followed by Dr. Ashley Morris and Lindsay Yoder, talking about homeward rebound, Vallisneria americana restoration in 2 New York waterways. So, with that, I'll stop sharing my screen. I'm going to introduce Bethany, and we can take it away. Bethany, if you want to share your screen. So Dr. Bethany Bradley is Professor of Biogeography and Invasion Ecology in the Department of Environmental Conservation at the University of Massachusetts in Amherst, Massachusetts. Her research has a strong focus on terrestrial plant invasions with a goal of understanding how invasive plant distributions, abundance, and impact vary spatially. Bethany also leads the Northeast Regional Invasive Species and Climate Change or Risk Management Network, which addresses the question of how we can better manage for upcoming biological invasions in light of climate change. Take it away.
Thanks, Kathy. And yeah, thanks so much for having me. I am starting all presentations this year, just noting that all of the research that I'll be talking about today has been supported by federal grants. Those have largely come through the Northeast Climate Adaptation Science Center, or NECASC, which is part of the U.S. Geological Survey's Ecosystem Mission Area. Funding for ecosystem science through the USGS and several other federal agencies has been appropriated by Congress, but blocked by the Trump administration, which makes it uncertain whether these programs are going to continue. The Northeast Regional Invasive Species and Climate Change, or RISC Network, is a flagship program of the CASCs, not just in the Northeast, but across the US. So that program is similarly at risk. All right, so I will maybe caveat everything that I'm going to talk about today with the point that I am a terrestrial invasive plant person. I'm a biogeographer by training, so I actually don't know that much about any specific species. I think about species as sort of points on a map. A lot of the invasive species and climate change research has happened on terrestrial plants or on aquatic, not plants. So what we're going to talk about today is going to focus on those two things. And the hope, of course, is that what we know about those two areas, terrestrial plants and aquatic vertebrates and invertebrates, might be relevant or will be relevant to the work that you're doing on aquatic plants. But aquatic plants and climate change is definitely an area that is underrepresented relative to the literature. All right, so we're all dealing with invasive species and aquatic systems. We know that that is a lot to deal with already. Invasive species are bad enough. And now we're saying we need to start thinking about not only invasive species, but what climate change is going to do to interact with invasive species. So what else does climate change add to this mix? It adds rising temperatures. It adds peak year precipitation, so both flooding and droughts within aquatic systems. more intense hurricanes, so higher levels of disturbance, loss of winter snow. And so combining all of those things with invasive species is definitely plenty of things to think about. If I can offer any silver lining for the interactions of all of those different things on our ecosystems, it is that oftentimes when we are dealing with invasive species, I would say not just oftentimes, but typically when we're dealing with invasive species, we are over, let's see if my laser pointer works, we are over at this end of the graph. As in, we are dealing with invasions that are already well established, widespread, have a lot of area occupied, and essentially we're trying to protect things from these widespread invasive species. That, unfortunately, is not a particularly cost-effective or ecologically effective approach to invasive species management. The much more cost-effective, ecologically effective approach is to stop them before they get there, before they have a chance to spread and have ecological impacts. So one tiny silver lining associated with thinking about climate change and invasive species is that that's where we are when we're thinking about climate change. We're thinking about the species that have not yet arrived, that are on their way here, and how can we prioritize those species to prevent their spread so that we don't end up, in this space with emerging invasive species. Another sort of silver lining here is that we know that as we learn more, we can better manage and help ecosystems to adapt. This is a graph that is from the Northeast Risk Network, and this graph shows responses of Risk Network members to the question, how many ways do you incorporate climate change into your invasive species management? So darker red on here indicates that respondents are saying that they're incorporating climate change in more ways. So more of these 5 specific ways that we laid out in the survey. And the graph shows that at longer term risk members, so those are the ones on the right, so people who have been thinking about this and learning about this for more than 3 years, are incorporating climate change into their invasive species management in more ways. Longer term risk members have also reported a higher level of knowledge about how climate change affects invasive species. So the more we know, the better we do. And we have in the Risk Network, we have recently had what we call a team up webinar, which is a presentation between both managers and scientists on hydrilla and the Connecticut River Basin. And we have an upcoming webinar on November 12th, so in a couple of weeks, on aquatic species movement associated with storm flooding from a large-scale water model that the U.S. Geological Survey is working on. So I'll leave a link at the end of this presentation, but if you're interested in more information about invasive species and climate change, Risk Network is a good resource for that. What I want to talk about today in terms of an outline of topics are 5 main things. And so I'm going to go through these in order. So the first one is how rising temperature interacts with invasive species, invasive plants especially. Second is novel introduction pathways associated with invasive species and climate change and range shifts, so the expansion of ranges of invasive species that are already here in the US. Fourth is climate variability creating disturbance, and last one is plant response to rising CO2. I'm going to do, kind of a sprint, I guess, through all of those different ways that climate change is interacting with invasive species, but we do have a cheat sheet. So this is available free to download on the Risk Network website. It's one of our research to practice papers, and we call this one Double Trouble. So what I'm going to go through are they're sort of labeled in order, although 5 and 6 I've jammed together into 5. So just a quick overview of what we're going to talk about. First are those invasive species phenology, so emerging earlier, staying longer because of extended growing seasons. Second are those novel introduction pathways. Third, range shifts and new introductions. Fourth, extreme events. And then fifth, the invasive species interactions with CO2. So starting with that first one of interactions between invasive species and climate change in terms of temperature warming. IPCC reports, every time one of these comes out, they try to make it even more clear in the language that there's no doubt whatsoever, unequivocal, that human influence has warmed the atmosphere, ocean, and land. So what does that mean for invasive species? So this is from a meta-analysis, and meta-analyses, I'm going to show you a couple of these. So a meta-analysis for ecologists is essentially where ecologists go out to the published literature, gather a whole bunch of studies that have already been done following a particular experimental setup. Like, for example, you have a native species that is under ambient temperatures and one under warm temperatures, and you're trying to understand whether it does better or worse under the ambient versus warmed. And then you get all of those studies together and you reanalyze them statistically so that you can understand an overall effect size. And an effect size in a meta-analysis, 0 essentially means that there's no significant difference from, or there's no significant change between those two treatments, warming and not warming. Positive, in this case, means that there is a positive effect associated with warming. Negative, in this case, means that there's a negative effect associated with warming. So this is a meta-analysis result. The aquatic one, which is outlined in red here, is 41 total aquatic studies. So 24 of those were for native species and 17 of them were for non-native species. The non-native is kind of the gray triangle and the native is the black circle. These are primarily not plants. So 95% of them were not plants. So they were mostly invertebrates and some vertebrate species. And generally what you're seeing here is that native aquatic species, so not plants, perform poorly with increased temperatures, whereas your non-native species don't really care one way or the other. When you compare those two, compare between the non-native and the native, you see that the difference between those effect sizes, so basically taking this minus this, is strongly significant in this case, and that you have a really positive benefit for the non-native species. So this is non-native minus native. So you have a positive benefit for non-native species when you have warming temperatures in aquatic systems. And so what might be causing that? One main reason is because warming causes a lot of physiological stress to aquatic organisms. In terrestrial environments, a lot of times organisms can go and find some shade. They can find slightly cooler places to hang out. In water, that's not so easy to do, especially with a lot of our aquatic systems not having a whole lot of shade or having barriers to passage between places. So this is just an example. This is for actually a marine fish in Europe, but illustrating that warming creates physiological stress in fish. So TC here is the critical temperature, and above that critical temperature, there isn't enough oxygen for aerobic metabolism, which means that the fish can basically no longer breathe. If it gets too hot, you end up with this very low dissolved oxygen content. That's especially true if waters are stagnant and not moving, because there's no interaction with the air. And native species are generally not adapted to those conditions, so they die. So loss of native species then creates a disturbance that creates an opportunity for invasive species to later gain a foothold and eventually potentially invade. Here's another meta-analysis. So this one is a meta-analysis that's specifically looking at plants relative to elevated temperature. You can see the little numbers in parentheses are the numbers of studies. So there were 31 studies on native species shown in blue, 20 studies on invasive species shown in red. This is for plants, but it's almost exclusively terrestrial plants. And what you see with terrestrial plants, at least, probably this is going to be true for a lot of certainly wetland plants. I don't know about, free-floating macrophyte type of plants, but certainly for wetland plants, that should also hold true, that invasive plants perform significantly better under elevated temperatures. And so why might that be? One mechanism for this is a priority effect. If you spend any time in the forest, which I imagine many of us do, then you're probably familiar with some of these invasive understory shrubs, so buckthorn or barberry, shown on the right. And what you can see in both of those cases is that the invasive understory shrubs are green, while the forest canopy has no leaves. So this is an early spring image of a northeast forest in both of these two cases. So one thing that invasive plants tend to do really well is to be kind of the first out of the gate. So they're ready as soon as it's warm enough. They're starting to germinate and grow as soon as the temperature is suitable. Whereas a lot of our native plants, and this is actually a big unknown across the Northeast, but what we think is that a lot of those native plants are not responsive to temperature, but are instead responsive to daylight cues. So they're waiting until you've got the right length of day, and then they're greening up because, historically that has worked well for them, but with rapid climate change, not working so well anymore. So priority effect basically means that by the time that native plant has germinated and has started growing, your invasive plant is already plantzilla, on top of the native plant, and is able to easily outcompete it and crowd it out. In aquatic systems, I think that knotweed is probably a good example of this type of phenomenon. We also see, so this is a consistent trend, not just for a couple of different species, but across invasive, again, these are mostly terrestrial plants, where these investigators at Boston University looked back at flowering times that were recorded from herbarium records all across New England that dated back about 100 years, so to the early 1900s. And what they found was that invasive plants are growing earlier consistently, and that they're showing this strong shift in phenology over that 100-year period. So if you look at the second one, native versus invasive, the invasive plants on average have shifted about 11 days earlier in terms of their flowering time relative to the native species. And this is specifically an invasive species phenomenon. It's not just native versus non-native. It's specifically those species that are having an impact. So essentially, invasive plants, because they're able to grow earlier, those are leading directly to their ability to spread and have ecological impact. So take-home point on this first one is that invasive species take advantage of these longer growing seasons, especially earlier spring, by emerging early, and that increases their ability to compete and also to have ecological impacts. And second, that native species, especially in aquatic systems, really can't take these increased temperatures, and in stagnant waters especially. Second interaction between invasive species and climate change are new introduction pathways. This is a map and a graphic from the US Global Change Research Program. That program, unfortunately, was canceled earlier this year, so we're not going to have much beyond 2024 in terms of data. But what you're seeing here is the extent of Arctic sea ice going from the late 70s to today, and you can see a strong downward trend in the extent of Arctic sea ice. So in my lifetime, we've lost more than 1 million square miles of sea ice, going from about 2.7 million square miles in the late 70s to 1.7 square miles a couple of years ago. So what does that mean for introductions? What it means is that the Northwest Passage that explorers were looking for, back hundreds of years ago, that Northwest Passage is now open for several months during the summer, which means that you can have direct introductions from temperate East Asia over here, coming through this way, and directly into around there someplace, the ports of New York. currently, if we're bringing plants or materials or anything else from East Asia, those materials are coming into ports of LA or someplace on the West Coast, and then they end up on a train or a truck or something like that to get to the Northeast. Which, if you are this might be more relevant for forest pests, but if you are a forest pest, for example, or any kind of pathogen or insect, getting on a truck from LA is really hard for you. If you're a temperate forest pest, those are likely to die. But if you're introduced directly into New York forests, then you might be a lot more likely to survive and affect our ecosystems. New species. So the first one was accidental introductions through that opening of the Northwest Passage. The other thing to bear in mind is that new intentional introductions are happening all the time. This is a map showing major trade partners for nursery plants to the US. So these are all countries of the world that, at least as of 10 or so years ago, we were importing a lot of ornamental plants. So those include wetland and aquatic plants, nursery plant, live plants for planting, basically coming into the US. The red ones on here are ones that were relatively new and sort of expanding quickly, and the blue ones are long-term established trade partners. So why are new introductions potentially a problem? One big issue related to climate change is that new introductions of nursery plants tend to be warm adapted. So this example is actually of expanding the range of a species that's native to Europe. So it's native in this red part in Southern Europe around the Mediterranean. And all of these green dots, which you can see if you squint at it, they go all the way up here. Those green dots are locations in Europe where nurseries are selling this Saponaria species. This is actually kind of an unusual example of a native species, native to Europe, being expanded north. Actually, most of the nursery plants that we sell, that are sold around the world, are non-native or introduced. So they are actually, like this species, coming from Europe and being introduced into warmer regions within the US. What this means is that we are already expanding the ranges of species that we select for gardening. It means that we are introducing them into places where they're going to thrive in the future. With climate change, we don't do a great job of doing that for native species. All right, so we've got all these new species coming in. What's risk associated with those newly introduced plants? Historically, risk associated with newly introduced species has been assumed to be relatively low. This goes back to what's called the TENS rule of invasion ecology, which was first published in 1996 and has kind of become a pretty often cited and you referred to rule for what risk looks like from invasive species. And the idea behind the TENS rule is that if you have, this is 100 little boxes, if you introduce 100 boxes, the TENS rule would say that 10% of those would go on to become established. So having a self-sustaining population outside of human cultivation. And that of these 10 that become established, 10% of those would go on to be invasive. So about 1 of 100 species that get introduced, according to the TEN rule, would go on to be invasive. The problem with the TENS rule is that it started out, looked at a lot of introductions that were accidental, but we know that when we look at invasive plants specifically, most of those introductions are deliberate. They're intentional introductions for ornamentals, largely for soil stabilization, other examples of us moving them around, which means that we actually want them to successfully, whether it's successfully thrive in our gardens or introduced into ecosystems in the landscape, this ends up being a lot higher. So more recent analysis of rates of establishment for introduced plants puts that number, rather than 10%, at more like 60%. Again, because we probably want those species to thrive or survive, whether it's in our gardens or in the surrounding landscapes. And then another recent analysis recalculated the proportion of established plants that are recognized as invasive across global ecoregions, and found that the number of the ones that are established, that should say invasive underneath, that go on to become invasive is more like 20% rather than 10%. And so from an overall visioning of risk, this means that it's actually more like 1 in 10 of introduced species will go on to become invasive rather than the 1 in 100 that we previously thought, which means watch out for new species. So here's a example tool to address some of these unknowns, which is called horizon scanning. And horizon scanning doesn't have to be used just in the context of climate change. It's a great tool of just trying to scope out what's around and what we should be worried about next. There are a bunch of different methods associated with horizon scanning. This is an example of a paper that was done for invasive plants in the state of Florida. So one example method, a lot of it just involves getting a bunch of experts in the room and looking at big lists of plants, and thinking about which ones are likely to establish where you are and which ones are not, and then narrowing those down into which ones are both likely to establish and also have pretty big impacts. Florida's been actually at the cutting edge of a lot of horizon scanning, largely because Florida doesn't have states to their south to look to in terms of figuring out what's coming next. And so they have to be more broad, to looking at nursery trade or looking at other species that are known to be invasive outside of the US. So a take-home point on this is that climate change is creating novel introduction pathways, including that Northwest Passage, and that intentionally introduced plants tend to be warm adapted, which gives them a big advantage in the context of climate change. Third interaction are interactions between range shifts, existing invasive species shifting their ranges in response to climate change. The Northeast, where we are, is a hot spot for future plant invasions. So all of these red colors up here in the Northeast and the upper Midwest indicate that up to 100 new plants are likely to, where they are currently in the Southeast or the Mid-Atlantic, expand their ranges north because temperatures are warming with climate change. We have done a fair bit of tool development to try to help identify these range shifting tools. So some work led by Jenica Allen in collaboration with the folks at the University of Georgia who run the Early Detection and Distribution Mapping System, or EDMAPS. They have a tool on their website, this range shift listing tool, where you can either at the county or at a state level generate a list of species that are not yet present, or at least weren't yet present as of a few years ago when we were putting this together, not yet present in that county or the state, but are projected to be suitable for your county or your state by 2050 with climate change. And at this point, we've done a lot of impact assessments on top of all of those range shifting species. So basically for every state in the Northeast, and in this case, our Northeast goes from Virginia to Maine, we've completed impacts assessment for all range shifting invasive plant species. And this is just an example. This is from the Risk Network pamphlet, highlighting some range shifting invasive plants that are likely to come to southern New England states. So New York, Massachusetts. I know that New York isn't in New England, but for convenience sake, we pretend it is. New York, Massachusetts, Connecticut, and Rhode Island, any of those different states. There were about 100 range shifting species that would come into those. And of those, we identified, I think, 8 or 9 species that have known high impacts in states in the Mid-Atlantic or further south. Here's a couple of wetland examples. So these shouldn't be too much of a surprise, I imagine. Arundodonax and Ludwigia grandiflora water primrose. So one question is, why are we worried about invasive species expanding so quickly? The main reason is because we can see across the board when we look at the rates of movement of uh, non-native and invasive species that they are successfully keeping up with climate change. So they're moving fast. So what this graph is showing is, and this is on a log scale. So as you get taller, you're like really high in terms of your spread rate. So this is how fast on average species are moving. This is all taxa, not just plants. Spread rates from 1 kilometer a year, 100 kilometers per year, 1,000 kilometers per year. Native species, there were 247 native species that we were able to find information for. And on the whole, they were moving at a spread rate of 1.74 kilometers per year, which sounds, slow, but not too bad. The number that we need to compare that to, though, is this dashed line. So this dashed line is 3.25 kilometers per year is how fast the average species needs to move to keep up with the current pace of climate change. That's called the velocity of climate change by climate scientists. So basically, if you're above this line, you are doing a good job. You are likely to persist under warming temperatures. If you're below this line, you're falling behind and your populations are likely to decline with climate change within parts of your range. The most straightforward comparison is between native spread and non-native spread. So our non-native species, these are ones that are moving within the introduced range. The average speed is 35 kilometers per year. So no problem. They're moving 10 times faster than they need to be. This difference actually is pretty consistent across different taxa. It's especially problematic for plants. So this dicots over here, if you look at plants, we didn't have as many studies for monocots, but about the same pattern. Dicot plants are actually moving at an average of 0.1 kilometers per year. And in aquatic systems, especially where there's, it's really hard to jump between, especially moving north, it's hard to jump between different aquatic systems. They are moving really slowly, whereas all these plants that are non-native are keeping up just fine. And so why are those non-native plants likely to be keeping up just fine? Again, this goes back to introduction of ornamental plants that are home gardening, largely introduces non-native species. This is from a recent study of about 330 nursery catalogs from across the Northeast. EF are eastern forests. So these are 985 species. These are the native species to eastern forests. There are some species that were not native to eastern forests. So these are ones that are coming from like the Pacific Northwest. But the real big one to pay attention to is that 63% of ornamental plants sold actively in the Northeast are non-native. Of those, about 1,000 have not yet established outside of cultivation that we know of. 555 have, and 283 are already known to be invasive. In fact, about 61% of invasive plants are still for sale. These are all terrestrial examples, so apologies for that, but some of the best examples that we have are the most commonly sold examples are miscanthus, silvergrass, barberry. Kogan grass is actually on the federal noxious weed list, still sold by 33 vendors in 17 states. So key point here is that continuing education about our landscaping choices is really important, not just for terrestrial ecosystems, but also for aquatic ecosystems. So people are planting plenty of things in wetland areas or adjacent to wetland areas. They may well be planting beautiful flowers in ponds as well. This is not just a terrestrial phenomenon. So for example, we don't have water hyacinth on this list because the risk does not yet expand to the Northeast by mid-century, but it's only a little ways to our south and temperatures are warming pretty fast. So even water hyacinth in maybe our grandkids' time is likely to be invasive up here. This is another risk resource called Do Not Sell, and a list of ornamental plants to avoid with climate change. There are a lot of example species in here that are like this one. This is a shrub called Beal's barberry, Mahonia bealii, where the species, in this case, all these hash marks down here are where the species is identified as currently invasive. So the currently invasive range. The red without the hash marks up here, including a whole bunch of New York and coastal New England, are areas where future invasion risk is high. And the little triangles, which you can kind of see here, there's one in New York too, those are nurseries that are actively selling this species. We see this with lots of different species that we are already facilitating the movement of non-native and invasive species to allow them basically to be pre-adapted to future climate change. This particular resource also has some native alternatives for each of the highlighted species that we've identified there. So take home point for this one is if we plant it, those species are going to be there in the future, and they're going to take advantage of those warm temperatures pretty much as soon as it's warm enough for them to start spreading and having impacts. Fourth interaction is the interaction between disturbance from extreme events and invasive species. These graphs are for frequency of hurricanes, so total numbers of hurricanes, and intensity, so the strength of those hurricanes in the North Atlantic since 1970. So 55-year record, 54-year record, and both of these. Pre-satellite is a little bit less accurate. Satellite record kicks in the mid 90s, and then it's more accurate over there. But these lines are general trends over that 50-year period that we are seeing more hurricanes in the North Atlantic, and that the strength of those hurricanes is increasing over time. Invasive species are well known to thrive when you increase disturbance. So we talked about that priority effect early on, right? If invasive species grab a foothold early, they can then usually outcompete other species they known to be disturbance responsive. That we may well end up with, okay, if we knock back, for example, these are examples from forest, of course, but you can imagine this for aquatic systems as well. If you knock back a lot of species, native species within those systems, then you can quickly transition in a disturbed landscape to a species like kudzu, for example, or knotweed is another good example that grows really fast and is able to take advantage of that disturbance. So knotweed is a good example of a species that's well known to spread after flooding events, like from hurricanes or even just the peakier precipitation that we see with climate change. Another thing that's associated with knotweed is not just its spread after floods, but an interaction between knotweed and erosion and flooding events itself. That knotweed is known to have roots that aren't super deep, so that if you end up with a lot of rainfall or flooding, you can have entire sections of knotweed basically slough off, right? So that facilitates knotweed spread. But the other thing that it does is it leaves a section of soil in your riverbanks that has no vegetation on it at all and can be easily eroded, increasing the erosion on those water edges. So take home point here is that climate extremes are creating novel disturbance, that novel disturbance harms native ecosystems, and harm to native ecosystems creates an opening and an opportunity for invasive species. All right, and then the last piece of interactions between invasive species and climate change is specific to plants, and that's plant interactions with rising atmospheric CO2. This is a graph from the late 1950s. I think it was 1957 that this was put in at an observatory at the top of the Mauna Kea, I think, volcano in Hawaii. They put this at basically as high as they could get, as far away from human development as they could get, so that they're generally just measuring what's going on in the total atmosphere in terms of CO2 and not being affected by, like changes in the cities or something like that, or agriculture really close by. So this is a pretty good measure of total atmospheric CO2 that has gone up, again, in my lifetime from, low 300s, well past 420 at this point in 2025. This little sawtooth pattern, if you're not as familiar with these types of graphs, the sawtooth pattern is actually caused by plants on Earth. That most of plants on Earth live in the northern hemisphere because there's a lot more land in the northern hemisphere. So you end up with essentially boreal forests and temperate forests in northern latitudes, greening up, and then what they're doing right now in the fall, browning down. And in the southern hemisphere, you have mostly ocean as opposed to land, and so you don't have that same effect in the southern hemisphere, which means that the northern hemisphere drives this little sawtooth pattern of the line goes down in the spring when all of those plants are starting to green up and, like start sucking in CO2. And then the line goes up where we are right now, where the plants are dropping their leaves and no longer absorbing CO2. All right, so plants generally do better with higher CO2, that trees have upwards of 50% increased growth, crops upwards of 35%, shrubs, flowers, 20%, 15%. On the whole, we see a greening of ecosystems with more CO2. The problem with this is that those averages are associated with only a couple hundred, maybe, total plant species that we have any information for. And most of them are staple crop species or species that are native to the US, where we like to study these types of things. We don't, this doesn't encompass kind of the variation between different groups of plants and what might happen. This image is actually from a website that was sponsored by ExxonMobil and the Heartland Institute that described why CO2 emissions are green. That type of information actually has gone back into a lot of executive orders this year, and some of the rulings, for example, for the EPA trying to roll back emission standards within the US because of greening. Problem for invasive plants is that when you have rising CO2, this actually gives them a big competitive advantage relative to native plants. So this is that coming back to that same meta-analysis that we showed way at the beginning. There were 45 studies of native plants. They do better. So they have a positive effect with rising CO2. 46 studies of invasive plants. They do even better with rising CO2. So this creates a competitive advantage. And the reason behind this is probably because invasive plants tend to be weedy, fast-growing resource hogs, basically, and CO2 is a resource that enables faster growth. So if you like to grow fast, adding CO2 is good for you. Another challenge associated with rising CO2 is that invasive plants tend to be harder to kill with herbicides with elevated CO2. These studies have only, to my knowledge, been done on a handful of agricultural weeds and not necessarily applied to environmental, ecosystem invasive species, nor do I know what the results are likely to be in aquatic systems. But it does appear to be consistent with agricultural weeds, that what you're looking at here, this is for Canada thistle. On the left is under ambient CO2 back in 2004, where they planted a bunch of Canada thistles where all these little markers are, sprayed them with the recommended amount of herbicide, I think Roundup in this case, and mostly got them, so one hardy individual there. This one on the right, they planted those same Canada thistles with all these little markers, sprayed them with the recommended same amount of Roundup, and did not get them at all. And they think that, so one possible mechanism for this, or two possible mechanisms. One is that if there's more CO2 in the atmosphere, then plants don't have to have their stomata open for as long, right? To capture the CO2 that they need for photosynthesis. And so that might reduce the efficacy of some herbicides that need to be absorbed through the stomata. Another mechanism in this case for terrestrial species is that these species were found to put a lot more biomass below ground into their roots with increased CO2. And so that allowed them to recover after that treatment. So, take home point is that aggressive plants really like rising CO2. Okay, so just to hit all of that outline of topics one final time, we talked about rising temperature favoring invasive species. We talked about novel introduction pathways, climate change and rain shifts, climate variability and those extremes creating disturbance, and finally, plant response to rising CO2. What can we do about all of that is the topic of a whole other seminar. So I would direct you that we have another one of these management challenges resources on the Risk Network website called Taking Action, which is from a couple of years ago, but is a nice synthesis of what people are already doing to try to mitigate some of these emerging interactions. That we're also, through the Risk Network, in the midst of aggregating a database of from about 60 different invasive plant lists across the Northeast to try to come up with a single list of invasive plants under both current and future climate. So you can check back on our website or sign up for our listserv. Feel free to do that to hopefully stay up to date with what's here and what's coming. And I will leave it there. That's the Risk Network website. Please feel free to join or check out some of the resources there. That's where all of this information comes from. All right, I'm going to stop sharing.
Thanks, Bethany. So next up, we have Dr. Ashley Morris and Lindsay Yoder talking about homeward rebound, Vallisneria americana, restoration in 2 New York waterways. And Ashley Morris works as the New York State Aquatic Invasive Species Coordinator for Region 3 of the DEC. She received her doctorate in ecology and evolution from Stony Brook University, where she studied southern pine beetle interactions. She has worked as the Region 1 Long Island AIS coordinator for 4 years before moving up to the Hudson Valley, now overseeing the Region 3 AIS strike team, which focuses on monitoring, management, and research of invasive species in the lower to mid-Hudson Valley, utilizing a variety of techniques to detect and manage invasive species and restore native habitat. Lindsay Yoder is the Region 9 Aquatic Invasive Species Coordinator for DEC. She works in partnership with New York State's Water Resources Institute at Cornell University, where she focuses on aquatic habitat assessments, invasive plant control, and native plant restoration throughout the Lake Erie and southwestern Lake Ontario watersheds. Together with her field team, the program tackles both small and large projects, including the Erie Canal Hydrilla Control Project, the Niagara River SAV Mapping Project to characterize aquatic vegetation both above and below Niagara Falls. She began her career in the field of aquatic plant management in 2017 with New York State's Lower Hudson Partnership for Regional Invasive Species Management before moving to Buffalo in 2022 for her current position. She has a bachelor's in fisheries and wildlife sciences from Oregon State University, where she specialized in aquatic ecology. All right, ladies, can you share your screen and please get started? Thank you.
Sounds good. Thanks, Kathy. We do have separate presentations, but I'm going to go first. Hopefully everyone can see that. There we go. Okay. As mentioned, we're going to be talking about our restoration efforts happening across New York. And so I'm focusing primarily on tributaries of the Hudson River, and this also spreads across New York to where Lindsay works, and she'll be discussing that. But first, we're going to tackle the plant that we're talking about today largely, which is Vallisneria americana, or water celery, or eelgrass, or any number of common names that it has. So why Vallisneria is our focus as sort of our primary restoration material? Well, it has a growth habit that establishes dense plant growth through stolons, which are sort of the runners, sort of like strawberries. They'll produce asexual smaller plants, and they do this very quickly throughout their growing season, and they can establish a really dense mat or bed of Vallisneria. And this bed can also prevent the growth of other invasives, sort of like what Bethany was talking about with invasive plants being able to outcompete natives. Vallisneria, if it has the chance to establish itself, can outcompete some invasives. Vallisneria is also perennial, with the plants being able to establish from roots from previous years, as well as in some locations, but not necessarily in much of New York, it can be evergreen. In my locations, at least, we do see most of the vegetative material die back annually. But they do have that capability. We might see more of that as climates continue to warm. Vallisneria also produces both turions, which are their asexual winter reproductive bud, as well as producing sexually using seeds. And both of these materials are pretty easily able to be propagated in lab environments and greenhouse environments and in the field as well. So that's also really important. We had to pick a plant that was going to be able to be propagated so that we have plenty of material to put in the water. And Vallisneria is also highly adaptive to a lot of different environments. And so Lindsay and I work in opposite parts of the state. We use information that we've gathered from various different parts of the country. This is a really well-studied plant, and it's able to establish really well in some slow-moving water and still water. And it even has some salinity tolerance, which is really important for me working in the lower Hudson. But this also establishes why it's so important to use material that is local to where you're working, as they are really highly adaptive, but that means they're highly adaptive to where this plant is growing. So Vallisneria's value as a submerged aquatic vegetation in general is that it can provide many ecosystem services, that as we talk about improving habitat quality through the removal of invasive species, we want to be able to put plants back into the environment that do these services and reestablish these services for the other organisms that are there. So SAV is really important for dissolved oxygen. The metabolism of plants releases oxygen into the water, as opposed to a floating or emergent plant where a lot of the oxygen gets lost into the atmosphere. It is also really prolific habitat and food for fish and invertebrates. A lot of young fish will use Vallisneria as nursery habitat, hiding from larger fish, as well as just food in general. Vallisneria and its pretty highly established root system can also help stabilize sediment, which can increase water clarity in some areas. And so that's also really important, as once it gets into the environment, it can also help allow that environment be better for other native plants that maybe are a little less able to produce really hardy root systems. So getting a little bit more specific into what I am doing in the Croton River. So the Croton River is a tributary to the Hudson. It's in the lower part of the Hudson. It's in Westchester County, so it's all the way down in southern New York. What was interesting about the Croton River in general is that in 2016, we found hydrilla in the Croton. It was treated using a chemical treatment that we're not really going to focus on largely, as it sort of predates my time in this position. But it was treated for several years. The hydrilla was effectively eradicated from the Croton, which was really great. But then, the other side of our invasive species management is being able to reestablish what was there before, both before the invasive species had a chance to decrease the natives, and also because this was a chemical treatment, it was important for us to make sure that we preserved the native biodiversity in the area as well. So in 2014, pre-treatment, All of the species that were present in the entire extent of the croton were surveyed. And at that time as well, the Vallisneria was collected from that local population, again, to preserve that local ecotype. And they were kept safe in a lab environment until post-treatment. So at that time, we were able to take those plants. Well, Dr. Engelhardt did this for us in her lab in Maryland. She grew them up, she genotyped them, so we know the diversity that we have. And then we also know where they were using these maps. So that's sort of what happened before. And then now, starting in 2023 is when we started our restoration of the river itself. So this is the map of the extent of the river that we're dealing with and trying to restore. You can see sort of that big blue area to the left of that map is the Hudson. So this river is tidal, which is really important. As we're discussing this, it's tidal. It's a bit saline as well. Again, so Vallisneria is a really good choice for us in this river environment because it's so plastic in what it's able to withstand. In 2023, our idea was to plant small packets of Vallisneria in a whole bunch of places, just kind of to try to get populations started in many different locations. So you can see on that map there, the little orange dots are where we survey annually for the plants within the river, and we've been doing that since 2014. And then the green dots overlaying that are where we tried to put in restoration material in 2023. And that material consisted of 5 tubers placed in cheesecloth bags that weren't really allowed to grow for very long in a stable environment. We pretty much placed those tubers in those bags directly into the river as soon as we got them in June. So you can see there that the leaflets are maybe an inch and a half long. They really didn't have a ton of time to grow in a stable environment. We then staked them in the river in June. You can see that we use these sort of construction stakes, driveway stakes. We skewered the bags through those stakes. We tried to put on some construction fencing to keep the plants safe from herbivory, but it didn't really hold up incredibly well throughout the season. The tides really moved it around and it fouled up really quickly. Because the tributary is very bioactive, we get a lot of really interesting living creatures that end up settling, barnacles and stuff like that, end up settling on a lot of the stuff we put into the croton. So something as structurally complex as that hardware fencing really didn't work out super well. So none of our plants in 2023 actually survived throughout the entire season. Germination was relatively low, never really exceeding more than 20%, and the cheesecloths degraded before the roots planted. So really successful season. So in 2024, our methods shifted. I did a bit of reading on what some other people did when they struggled to get some establishment within river systems. We focused on what some people have been doing in the Chesapeake Bay region. And we decided to shift to, instead of planting little single bundles of a couple of turions, we decided to focus on larger patches in fewer locations. So really putting in a lot of effort into the plants themselves and just focusing on a couple of locations. So we chose 2 locations, again, based off of that 2014 map, where there were high amounts of Vallisneria in the past, one of which was further up the river near that island, and the other of which was closer to the mouth of the river, just to see if there were differences in those locations and to see if, maybe we can start for further away areas and start building in toward that middle part. So basically what we did was we wanted to create little pieces of turf to put into the river. And so how we did that is we took, instead of, 4 to 5 turions, we took 16 to 25 turions and we placed them within a burlap bag that we filled with topsoil. And then those plants were able to grow in a greenhouse environment for 3 months. So you can see here us placing that little tiny green turion in between those tweezers there into the burlap sacks. We then moved the burlap into these tanks and allowed them to grow. And so this is what they looked like when they were about to be placed into the water. It's, obviously a really, really stark difference from those little cheesecloth bags that maybe had an inch or two of leaf material. These plants have produced roots, they've produced stolons and runners, and some of them had even started to flower at the point that we were going to move them into the river. So this was a really big difference. Our tank setup also consisted of an aerator. Um, we were really grateful to be able to use Cary Institute of Ecosystem Sciences greenhouse. They loaned us some space. And so, we have this greenhouse that is able to kind of keep the temperature relatively stable. The plants do really well in the heat, so we didn't have to worry about that too much, but it's really making sure that they stay warm enough. So in August, kind of almost the same as what we did in 2023, we took driveway stakes and we staked the burlap sack into the river at those two different locations. And we noticed from 2023 that the plants that did germinate also disappeared very suddenly after that. So we were pretty sure that herbivory was something to be concerned about. So we placed these herbivory cages over our patches. Importantly, to note, because of the environment that we work in, our cages have to have roofs because we're working in a tidal environment where, we place these plants down, and if it's low tide, maybe the plants are in 6 inches of water, but that means all the way at high tide, they could be covered in 4 to 5 feet of water. So we really had to be sure that the cages were going to be able to withstand that amount of water movement per day. And you can see on the right there, we really wanted to show that the plants completely filled out that entire cube of space. So it worked really well this season in 2024. But what we did notice when we came back at the end of the season is that all those plants were basically lawn mowed out the top. We didn't see any plants out the sides of this cage, and we didn't have any leaflets growing out the top of it. And it looks like they had been grazed. We also pretty regularly see very large carp in that area. And so this really, again, continued to verify the importance of putting in herbivory protection. But also the holes in this cage are large enough to where really small fry could still continue to use these plants as habitat while we establish a large enough population to stand up to some of those grazing fish. So our overall results and takeaway from 2024 is that using this method where we planted larger amounts of plants in fewer locations, we saw way more success in the plants being able to stand up to the harsh environment that the river has. And so all the plants survived the summer. Specifically, the plants that were planted a little further upriver near the river island continued to flower and actually set seed. And hopefully those seeds, continued to establish new vallisneria in other parts of the river. We didn't go back and collect them because we had plenty of reserve material that we keep back in the greenhouse. So we also noticed that because the area near the river island was exposed to much lower sedimentation, it's not as close to the mouth of the river, they tended to do quite a bit better than the plants that were exposed to higher sedimentation further down the river. The plants that were almost in the bay of the Croton River kept getting pelted with really fine sediments as, again, the tides continue to go up and down. There were not a lot of other plants in that location as well. So there's nothing stabilizing that soil, and vallisneria really struggled to establish. So they survived the 2024 season, but when we came back in 2025, we were able to find the plants that were upriver, but we were unable to find the plants that we had planted downriver. They were not able to reestablish from tubers or turions back in that lower part of the river. Also, our herbivory cages weren't really made out of the right material for the Croton. The metal corroded really quickly, and so we had to, again, adjust our methods in 2025. So we were really happy with how the planting method worked out with us planting those larger patches of vallisneria turf. So we continued to do that in 2024 with us also focusing on 2 additional locations. We really wanted to focus on locations with high potential success. So we moved our 2 locations again further up, further downriver from our original island location, but further up from the one where they had really high sediment. So we're hoping to fill out the areas with low sedimentation first, and then work our way establishing higher amounts of root systems, hopefully kind of letting these plants naturally encroach into areas that are a little bit more challenging for them. So we did the same thing as we did in 2024, where we established the plants within the greenhouse and planted them in August. Also in July, we went back and put new plastic PVC cages over where we were seeing plant growth come back. Yeah, because importantly, so these 2 pictures on the side here, you can see that that top picture is what the Vallisneria regrowth from 2024 looked like in June. And that bottom picture is what it looked like when it was uncaged until July. So you can see that there is a reduction in density. Fortunately, we were able to put a cage over that and the plants did well the rest of the season. But we definitely needed to leave the herbivory cages on for multiple seasons to allow them to establish a more robust population. In addition, this year, we also planted empty expansion cages next to each of those 3 plots, hopefully to allow the natural protected expansion of the Vallisneria into these new cages. And what we found is that the plants that we actively planted within the cages did really well and they grew. But we didn't see any expansion of Vallisneria into the empty cages, but also interestingly, we also didn't see any expansion of invasive species into those empty cages either. So all of these locations that we're working with have pretty moderately dense milfoil in there as well, as it's also trying to reestablish after the treatment. But when we placed down those cages, nothing grew in them. So we are hoping that maybe we can use them as placeholders for restoration material, but we definitely have found that at this point currently in the Croton, we need to do active restoration and can't just, protect empty spots hoping that something will come in. And so overall, some of our larger takeaways from what we've been doing in the Croton for the past 3 years is a lot of this is trial and error. Is that, a lot of what's important we have seen for successive restoration is acknowledging that the environment is going to be different everywhere. You can't just read something that's going to work in one location and assume it's going to work in your location. As we did that in the beginning and stuff didn't really work the way that people said that it worked in other areas. And so there's no real size fits all solution to any particular restoration project, but instead, there's different kinds of environmental variables that need to be considered and adapted to and tested as you move forward in a restoration plan. So some of the things that we highlighted in our river that were important and probably are important in general in any kind of tidal system is that the Croton River had areas of really high sedimentation and turbidity. We had to keep into account the tidal movement, so there are large changes in depth throughout the day. There are also other invasive species in the Croton. There's milfoil and brittle naiad. There is decent populations of large herbivorous fish. And there's also moderate recreational usage and residential ownership all along the shorelines. And so again, throughout our 3 years of working at this site, ways that we've worked to adjust to each of these variables, some of which was just by looking around at what worked and what didn't in the years that we've been conducting this. So again, to account for the areas of high sedimentation, we just paid attention to what the plants looked like. We noticed that further down the stream, there are really no plants coming in right now, and the plants that are really browned out by having so much fine sediment on them. So we paid attention to that and paid attention to kind of where that line was. So identifying areas that were going to have higher success at this time. The tidal movement, we accounted for that with all of our restoration plantings took place at low tide, ensuring that none of our plants are exposed to air. Vallisneria, at least our Vallisneria, was really sensitive to being exposed to air. So we really wanted to make sure that at all times of the day, they had water over them. You know, we didn't want to have the plants going multiple hours just on dry land. But we also you can't plant them too deep because then they won't get enough sunlight. So we had to find kind of that perfect area. And so that's why we plant at low tide, to make sure that that's kind of the lowest amount of water they're going to be exposed to. We pulled any invasive species that were in the area before the planting. We're hoping that at some point the Vallisneria is again going to be able to compete pretty well with the milfoil. But when it's first getting established, we wanted to make sure that we aren't going to run into any negative priority effects. We want the priority effects to work with us. And so we made sure that all that milfoil was pulled from around the area before we started. And again, like we're hoping in the future to take advantage of the fact that cages also sort of prevent invasive species encroachment. The herbivory cages sort of solved our herbivorous fish problem. Again, we're going to have to use them for multiple years before the Vallisneria beds get large enough to allow us to take those cages off. And we chose areas that were out of the main channel, and we made sure to place signs so that people knew what these cages were. We had a lot of people asking us if we were crabbing. And so we really wanted people not to mess with a lot of our stuff. This also had sort of the side benefit of, we chose a lot of these locations, like this picture here. Our plants we planted in this area, like all within the sticks, partially to have people not run into them. But then we also found out that a lot of the native plants did really well in these sort of naturally protected areas. So, areas that have tree falls and stuff like that, we started noticing more native plants growing in those areas first. And so our annual SAV surveys also help us identify locations where some of these plants might do better. So again, it was a lot of trial and error. And it certainly was a lot of trial and error that I did not do by myself. So I'd like to thank Megan Odell and Chris Donnellan, which were my tech and intern for this year. Dr. Kasia Engelhardt grew our original Vallisneria stock and kept it safe throughout all those years of treatment. So we really appreciated that. She taught us how to grow it within the greenhouse as well. I'd also like to thank Cary Institute of Ecosystem Sciences, as they gave us space in their greenhouse facilities. And that allows us to have native stock to put in to the river every year, as well as Dr. Catherine McGlynn for just her general support through the DEC and the Environmental Protection Fund, which funds a lot of our work. And I think with that, and that's my contact information if anybody's interested. And I can shift over to let Lindsay tell us what's happening on the other side of the state.
Great, thank you, Ashley. All right, let me share my screen. Okay, good. Can everybody see my screen?
Yes.
Great. Okay, so I'm really excited to share our work. I think you're going to find that Ashley and I have a lot of similarities, but a lot of differences as well. So pretty interesting to compare the two and see what's worked for both of us and what things are different. One of the biggest differences with our project is we are doing aquatic plant restoration alongside current chemical control of hydrilla in the Erie Canal. So the first thing I want to touch on, restoration, we have learned, is a very critical component of aquatic invasive species management projects. I think that has been a general understanding that if you're doing 1 to 2 years of chemical treatment for aquatic invasive plants, you can usually count on native plant restoration or revitalization like on its own. You might not necessarily have to go in and reseed those populations. But when we're dealing with these long-term legacy projects like the Erie Canal or like the Croton River, which was also a long-term hydrilla control project, those treatment processes in themselves create these disturbed open areas, which allow for potential recolonization of native species, but also of invasive species, either, a return of an old species or a new invader. And it, increases the risk of new colonization. So we have, realized that we need to really be focused on restoration, and for the canal, restoration alongside active control. We want to try and like sort of jumpstart that restoration process. So our legacy of the canal, we've had pretty successful control and reduction of hydrilla over the past decade, first by the Army Corps of Engineers, which spearheaded the project, and then the DEC took over the project in 2024. And historically, contact herbicides were used, which did a great job at treating the hydrilla, but unfortunately there were also non-target impacts to native species. So in 2024, we switched to a more systemic herbicide to try and combat those non-target impacts. And we also initiated concurrent restoration of Vallisneria to sort of, like I said, jumpstart that revitalization of native plants and potentially use Vallisneria as a, sort of biocontrol. As Ashley mentioned, Vallisneria is very good at outcompeting invasive plants. That's what we've seen in the Niagara River and other populations nearby. So we wanted to try and do that here in the Erie Canal after a failed pilot attempt in 2023, very similarly to Ashley. In 2023, we attempted to start a restoration program. We just did a really small pilot in the Erie Canal, and we did not see great success. We collected plants from the Niagara River and we planted those in laboratory tanks primarily to test propagation techniques for like long-term cultivation, as well as we collected those plants and planted them right away into the canal. And we found that the plants could be grown in a laboratory setting, but it would not be suitable for a long-term cultivation program simply because we can't mimic natural conditions that Vallisneria needs to senesce and produce turions. It's possible you could, if you had temperature control and you really went that direction. But as it stood, we really couldn't replicate the turion production that's needed to sort of scale up a large restoration program. And then in the field, we found that our plantings just failed miserably. We did not have nearly any survivors by the end of the season for a lot of different reasons. The planting method we used, we had some transplant shock, most likely. These were actively planted from plants that we had just collected. We used like a looser burlap that the plants maybe fell out of. We had these really small cages. So just not a very successful pilot project, but we learned a lot of lessons. The biggest one being that we needed to go into a greenhouse. We really couldn't stay in that lab situation. So we initiated our large scale program by partnering with the North Tonawanda Botanical Garden, which allowed us to acquire that necessary greenhouse space and expand our cultivation program pretty significantly, growing out our plants in larger vessels and utilizing a more natural, low-tech growing technique. So to start wild stock collection, we did that again once more in the Niagara River. We collected Vallisneria from two distinct sites that were very far apart, several miles apart in the river and on opposite sides of the river, just to sort of increase our likelihood of genetic diversity. We selected 40 to 50 plants from each site along a 50-meter transects using a designated protocol that we use to limit our disturbance to the natural habitat. Which I could certainly share with anyone who's interested after, but briefly, we just look for sites that have greater than 30% coverage of plants within 1 meter quadrats along those 50-meter transects. So here's some images of us doing that collection and what we look for within the quadrat, and as well as what we look for with the plants. We're really looking for plants that are anywhere from 2 to 4 inches tall with kind of weak root systems, just to improve our efficacy and reduce transplant shock when we're doing active planting. And those plants that we collected had a couple of different pathways that they went. The first was for long-term cultivation. So we brought the plants into our greenhouse, we decontaminated them using a pretty dilute bleach solution, which we then have to follow up with like a tap water conditioner or a dechlorinator. And the time that you rinse those in those solutions is going to vary based on the species that you're using. But Vallisneria tends to be a pretty hardy species when it comes to the decontamination process. We separate the plants by both site and specimen ID until genotyping can occur. And I will just give a caveat with the canal. We did not do genotyping right away, like prior to any Erie Canal planting. And that's just because we really wanted to get some plants in right away. We were dealing with an extremely low diversity community in the canal. We really wanted to get some plants in, and the canal is connected right, to the Niagara River where we were collecting from. So we weren't really concerned about that. But we did want to separate them out in the greenhouse until we could get that genotyping done. We planted the plants in single plants in 4-inch pots with unmodified topsoil, so no fertilizer. And we also topped it with sand to reduce turbidity in the tanks. We placed those plants in the stock tanks, and we fill that to the top of the pot. We just put enough water to just cover the plants until they get tall enough, and then we just keep gradually adding water to decrease the amount of distance that light needs to penetrate and get to the plants. Some of the extra plants that we collected were planted in larger containers just for like experimental observation and looking how they did in larger containers, as you can see on the bottom right image here, or bottom left image, when they're in these like larger buckets on tables. We just kind of want to see how they would do in that as opposed to in the tank, which was our primary method we ended up utilizing this year. So the other path that these plants went was into the field. So we still decontaminated them the same way using that bleach solution. And then we placed 5 plants in a 4x6 burlap bag. These are like just little drawstring bags, which were very convenient, with the same topsoil and a little bit of cobble just to add some extra weight. And we used disintegrating landscape stakes to install those in a grid pattern at a rate of 5 bags per 25 square feet or so within these constructed cages. And in 2024, we did 4 sites. We construct these cages using rebar covered over top with a PVC cage. We also conducted some macroinvertebrate sampling before planting so that we could do some sort of before and after. The after, which we have not done yet. We're waiting till we have better establishment, but we did do that in 2024. And you can see here on the bottom right, this is just what our bags looked like in 2024. So these really small, sort of like, small bags. Some of the monitoring we did for this, we measured our plants at the time of planting, followed by monthly measurements in the field and in the greenhouse to monitor growth, survival, and clonal reproduction. That, as Ashley mentioned, this plant produces runners and clones throughout the season. So we wanted to measure how many clones each of these specimens were creating. We also took some secondary measurements, dissolved oxygen, pH, temperature, turbidity throughout the season just to see how that might change. But these were really discrete samples, so not great at telling us much, but we still took them. The first round of clones that were produced were removed and placed into larger buckets on greenhouse tables to encourage growth and create a larger stockpile of turions that we could potentially use for the following season. So some of the results that we saw in 2024 for our greenhouse cultivation, these are just the 2 sites, 102nd Street and Buckhorn Island State Park. These are the 2 sites where we got these plants from. So you could go through all of this, but the main thing I want to highlight here is looking at the average height at the end of the season of these plants. You'll see that the 102nd Street on average was taller, produced higher, like longer leaves, but you'll see everything else was much lower, right? The total change in height throughout the season, or the rate of growth, was lower than the other site. It produced fewer clones and it produced much fewer turions. So what does that tell us? It tells us that that site maybe isn't great for long-term restoration producing founder colonies, which is what we're really looking for, because it just has a higher focus on like taller biomass, which isn't necessarily as important for us as it is like spread. We want something that's going to be able to really like spread out. But having the 2 mix might also be an option, but it's just something we'd want to focus on. We don't want to, and this is why we were doing this type of sampling, right? We don't want to pick something from one site that maybe doesn't do as well. So this was really helpful to help us determine like the sort of spread of plants that we want in our plots, and focusing on that too. And we also saw some, pollination and flower production. And then this bottom right is just what the turions look like after we harvest them, but we'll talk about that a little bit later. And in the field, we saw a much better outcome than we did in 2023. So this was really exciting for us. whenever I was really able to look at this data and compare it with the previous year, is pretty validating, that the work really paid off. Even in our worst sites, we still had 60% survival, and over double the growth. So it was still a very, very promising outcome. And also, just to touch on these two sites that did worse, the survival of 60 and 63%, that's kind of as expected. These sites were muckier, they had lower sunlight availability, like they were kind of in shady areas. So we didn't expect them to do very well. So they already did better than we thought. And yeah, just looking at the growth, this is from the Amherst site here on the bottom right at 41, 62, and 83 days. And you can just see just how much the Vallisneria started to fill out that cage just during the season. So overall, really great results. And in 2025, we did our first harvest from our greenhouse plants in February, which was really fun. We were not expecting our tanks to be, we expected there to be ice, but we weren't expecting there to be like 7 inches of ice that we had to dig through, and nor were we expecting some of our plants to still be green under the ice. But we had a closed greenhouse from November to February, so we really weren't able to get in there until then. But we allowed for a full winter for the plants to senesce or produce as many turions as they could. And we harvested from 78 specimens. We started out with 100, if you recall, but some of them died throughout the season. So we had 78 left, and we harvested from February to March. And all of those turions kind of went in different directions. We had a storage experiment. We looked at germination rates at 8, 12, and 16 weeks after being refrigerated, less than 50 degrees. Some of the plants we also replanted in pots for, again, genotyping or long-term cultivation. And then others were prepared for live planting, where we really wanted to plant from turions. Like as Ashley mentioned, planting from turions is going to be a little bit better than just sticking plants in a bag. You're going to allows them to, kind of root in place and just really establish themselves. So it's better to plant from a turion than it is a larger plant transplant. And that's just what those look like. We planted them in these bags and they start to sprout out. That was after just about 4 weeks. And there was a clear distinction in productivity between the 4-inch pots that we originally planted in and the larger vessels, which is pretty obvious. There was more space, more soil, whatever, better performance overall because it could produce more of those runners. So that's kind of where we looked to move into those larger vessels in 2025. We don't really use those 4-inch pots as much anymore. We also experienced some leaf melting and chlorosis in plants mid-season. We weren't sure if that was from chlorine or fluoride or light availability, so that was something else that we needed to look at this year. So in 2025, we expanded the plots at our most successful sites, which we're calling Cattail Marsh. Don't knock me for the name. I don't even know if it's cattail or a marsh, but that's just what we ended up calling it. And the Amherst site. So our additional plots, they match the characteristics of our previous successes from light availability and higher clay soils. And you can see the new plots versus the original plots in the background. The new plots were much larger because we really felt that we could scale up this season. And we used larger bags as well. We switched to 8x10 as opposed to the 4x6, just because, we could. We had more turions and more plants available. So that really worked out pretty well for us. We also created this signage that has QR codes so that people could learn more about our project. And we staked a subset of the plants so that we could measure them throughout the season again. Probably the most exciting thing this year was the resurgence of our 2024 colonies, which were meant to be founder colonies, and it worked. We had an incredible resurgence. I mean, even more than what we saw in 2024. They came back way stronger. Granted, we did have a very warm year, so that definitely helped them. But you can see we had a lot of community utilization of the plots from these frog eggs we found in June, and that was June 2025, not June 2024, sorry. But we found some frog eggs. We saw a lot of young of year fish using the plots as nursery habitat. We saw, unfortunately, turtles inside the cages, which we really didn't want, but they didn't seem to hurt anything, so I guess that's okay. And we did see spread outside of the cages, which, as you can see here at Amherst, we removed this cage in August because we felt that the cage was really hindering the growth of that area. So we went ahead and removed it. We removed the cage in Cattail Marsh as well. And I'll talk a little bit about that at the end. But yeah, you can see it spread out of the cage. So that was pretty exciting. So some of the results of the data from this year, obviously we had a higher overall growth rate and survival in the field. And that's, I think, attributed mostly to the fact that we planted from turions as opposed to larger plants that might have had some transplant shock. And we also established them early. We planted those sacks that we made in March. I think we planted them in mid-May. And we also had very warm temperatures this year in the canal. I mean, we had regular 80-degree water days. So that probably helped the native plants a little bit, helped the invasives as well, unfortunately. But we saw, 100% survival. We saw no mortality this year in our plots. We saw incredible growth and change in height throughout the season, more than we did last season. Average end height, it's variable from the previous year, but still really good. And in the greenhouse, we saw overall survival and growth in the greenhouse plants of the ones that germinated. And you could see the average growth. We can't do end height because we started from turions, but still really good, 16 to 12 or 12 to 16% growth per day, and pretty good survival between both of those. And also you can see an increase in the average end height at the end of the season from the previous year, probably again attributed to those larger containers. That's what I'm attributing it to. We did still experience the chlorosis and melting of the leaves mid-season, so I'm exploring that, which I'll touch on too at the end, but yeah, still dealing with that problem. One really cool thing about this project is our opportunity for community engagement. We've hosted several different workshops with kids and adults, interns. Kids really love planting aquatic plants, something I did not necessarily think anything of, but of all the kid groups I've been able to work with, they really enjoy it. So something to think about if you're looking to start a restoration program. It does make a really good community outreach event, and people are pretty engaged and interested in it. So looking ahead, some of the things we're considering, we're going to keep exploring additional species for restoration. We did also, I didn't really touch on it here, but we do also grow water stargrass and sago pondweed. Water stargrass produces mostly through vegetative propagation. It produces roots at nearly every single node, so very easy to propagate. And sago pondweed produces turions similarly to Balasnaria and some other true pondweeds. But we've been growing both of those, and we're going to keep exploring other potential restoration plants. We'll be conducting an analysis of the influence of the different genotypes on certain factors that would contribute to plant productivity. So looking at the relationship between dry biomass of Balasnaria leaves, number of leaves, number of plants on overall turion production, weight, length, and the relationship between weight and length on the plant performance in the next season. So just looking at some interesting analysis to do there. We want to really home in on our best genotypes so that we can start growing those out in larger vessels and reduce the need for all this individual specimen tracking. I mean, right now we're tracking anywhere from 70 to 80 different individual specimens, which we're treating as different genotypes just in case. But I'm sure that they're not different, a lot of them. So we are doing the genotyping, and we'll be looking at that in the future. Troubleshooting this chlorosis problem, which may or may not be influencing turion production, we're not really sure. So for fluoride, which we think is a culprit, we're going to be switching to a reverse osmosis filter. Right now we're very low tech. We just use tap water, and it really doesn't do much damage until halfway through the season, where we've been adding more and more fluoride all season. So we're going to switch to a filter. We think potentially the heat might be a problem for us, and that's just because we do kind of keep the water levels low during the early season or until the plants get very tall. And with some of these species that are shorter growing, the water stays low for a really long time. So the water gets really hot. So needing to fill the tanks to keep the water from getting so hot, we might need to put plants on risers to lift near the surface as opposed to having the water lower, looking at different, different things for that. We also want to move to a larger format planting over hydrilla beds, which are really close to our existing plots. We did that on purpose. But we want to switch to using like a full burlap sack, like a coffee bean sized burlap sack potentially, and just putting a bunch of them down right on top of beds and seeing if that can get us anywhere with some of those hydrilla patches. And then we'll be looking at some of these before and after field observations of macroinvertebrates, fish in and out of plots, installing some floating cages, which is a design that's being done by the Army Corps currently for more passive restoration of Vallisneria and also coontail and other vegetative reproductive species. And then not next year, but in the future sometime, a homeowner certified habitat program. The Erie Canal has dozens of homeowners, and there are some that have expressed interest in this, like having a certified SAV habitat in their backyard that they can take care of and kind of manage. So key takeaways, I'm going to try and wrap this up. I know I'm behind. But so for wild collection, collect as soon as temps allow. So if you're going to go out and collect once you can withstand it. So we go out in 60 to 65 degrees, collect those short plants, 2 to 3 inches with minimal root growth. Cultivation, you can get really technical or you can be very simple and very low tech. All you really need is tanks, tap water, some soil and buckets. You might not even need tanks. This is at Dr. Katya Englehart's greenhouse in Maryland. She just uses the buckets on the tables. Like she occasionally has some tanks, but this is really what she does, and it works great. You can harvest turions anytime after the growing season ends, but you need to bear in mind what you want to do over winter. Refrigeration can only last so long. You might want to do soil overwintering, or you can trick your plants into growing year round by moving them into a lab, which is what we do as sort of like a assurance or insurance plan. We do continue to grow some throughout the winter in the lab. We have a higher success rate and with survival and production in larger vessels or deeper tanks. And even with that higher success rate, if you're able to transplant new wild stock into laboratory settings before moving into a greenhouse, that's going to be better for you. So if you collect from the wild, grow it out in a really ideal environment first before moving it to a greenhouse, which is still can be really simple. We just have lights and some basic aerators in totes. And then similarly to pot size and greenhouse, the cage size does appear to make a difference for us. The plants didn't survive those single plant tube cages in 2023, but they're doing great in the really large cages that really give them room to spread. And then if you're able, if your site allows, like Ashley was mentioning, hers is tidal and it's very different. But for us, we can remove the cages and we haven't seen any issues really. So removing the cages after a couple of seasons might be beneficial for you because I do think that the cages can hinder the real spread potential of the plants. And again, all this work was conducted in basically lentic conditions. The canal is flowing, but not really. It's almost stagnant most of the time. So your mileage can vary depending on flow. So proceed with caution. And I just want to thank some people, my team, Dr. Katya Engelhart and Lindy Dodd, both of whom work on restoration projects and have helped me a lot troubleshooting and coming up with different ideas. And, I've really developed our methods based on their two programs and kind of like meshing them together and kind of seeing what works for us. Dave Conti at the North Tonawanda Botanical Garden and Dr. Kathy McGlynn, our fearless leader, as always, and the Canal Corporation and North Tonawanda Parks and Rec. So that I'm done, and now we can move on to questions.
Okay. Thank you both. That's great.
Thank you.
So let me share my little humorous slide. Oops. There we go. And then we'll take a peek at the chat box if I can figure out how to see it. Let's see. All right, maybe I'm stop sharing because I can't see the chat box. Hold on. Okay, that's much better. All right. So first questions in the chat. Let's see, we've got from Ken Wagner on the slide. This is for Bethany. On the slide showing the progression of an invasion, where do the economic return on investment ratios come from?
Yeah, thanks for asking that question. I actually went on down a little rabbit hole while while Ashley and Lindsay were presenting. That is from a study in Australia, actually. So I think we take the actual numbers on it with a grain of salt. But that I think there's plenty of good evidence for economic benefits of proactive prevention over the expenses of dealing with something when it's already abundant and widespread. The actual like numbers associated with those will vary by species and system and stuff like that. So that Australia study was one from 2008. It was actually done in Tasmania, and it was specific for agricultural weeds and the costs of preventing their introduction versus the costs of dealing with them after they're already affecting agriculture. So definitely one where it's going to vary a lot, but I think the invasion curve is pretty consistent across systems and species.
Great, thank you. Right, scrolling through. Next question from Elizabeth. Can you repeat, and maybe this is too late, but 63% of ornamental plants sold actively in the Northeast?
Yeah,
Okay.
61%. So this was from a study led by Eve Bury, who's now at the New York Botanical Gardens, and she and a UMass undergraduate student has spent the better part of a year Googling to find out what was available for sale. So essentially Googling the common names and scientific names and for sale to see what we could find. So that's not just in the Northeast, that's actually across the continental US. A small part of that is our species that, are unlikely to maybe ever become problematic everywhere in the US or in New York or New England. Like there are a handful of species that of fire-prone grasses, for example, that affect semi-arid systems in the Intermountain West, in the Southwest, and Hawaii that probably are not anytime soon going to be an issue in the Northeast. So there's a handful of those that, okay, legitimately, if we're planting some of these ornamental pennisetum or something like that, grasses in the Northeast, we might not need to worry about that particular impact. But the vast majority of the problem here is the fact that state regulations are extremely inconsistent and they're also reactive. So the stat that we had for consistency across state regulations is that on average, when you compare one state to a neighboring state, in terms of the invasive plants that are regulated between those two states, you would expect the overlap to be, I don't know, let's say 90% would be a good number that you would expect an overlap to be because there are going to be similar ecosystems and similar species that are sold. And the actual number is 17% overlap between adjacent states. So if you think about New York and New Jersey, for example, New York does a really good job of investigating emerging species that are likely to become invasive and recommending them for regulation and has funding and support to do that. New Jersey doesn't even have a regulated plant law, so they have nothing regulated whatsoever in that state. And those are kind of two extremes, but everything in between those exists. In Massachusetts, regulated plants are based on volunteer efforts. So if volunteers have time to do invasive plant risk assessments, then they might eventually make it onto a regulated plant list. Rhode Island has no regulation either. So it's just a huge patchwork out there. And that means that you end up with a very leaky sieve of what can and is sold across different states.
Okay, thank you. Let's see. And I had a question, and I think you answered it when you posted your Taking Action webpage, but I was just asking, how can the speed of movement of native plants and other native organisms be increased? Is there a way to facilitate this?
Well, yeah, and actually we just did a webinar a month or so ago about assisted gene flow too. So when Ashley and Lindsay were both presenting, a lot of historically, what we've thought about in terms of restoration, the target of restoration has been like what was there before the disturbance happened, or what was there before the invasive species was there. And that in the context of climate change is, short-sighted, I guess, in terms of trying to facilitate the movement of native plants with climate change. Right now, so the climate change that we've already experienced to date is the equivalent of moving about 100 miles to the south geographically. So that is plant communities that are about 100 miles south of us are the ones that are adapted to the current climate conditions where you are now, if that makes sense. And that's only increasing as time goes on. And so if you're doing restoration work, then thinking not only about sourcing, like when you're going to dig up some of those plants, if you're sourcing stuff in the fields, going further south is a good recommendation for assisted gene flow at this point. Going further south for species that might not exist in your location yet, also still a recommendation for climate adapted conservation. So that's how we help native plants to respond in the same way that invasive plants already have.
Thank you. Let's see, just scrolling through here. Oh, from Margot, we have what percentage, this is for Ashley, what percentage of the original Vallisneria beds were lost during treatment?
So yeah, I saw this question and I was not looking forward to answering it because it's complicated. There currently aren't any persistent Vallisneria beds in the Croton post-treatment. They seemed like they were kind of already on a decline from our 2014 to 2017 surveys, because we did a few years of post or pre-treatment surveys. Vallisneria has this weird tendency of kind of moving around. The treatment definitely knocked them back pretty hard. The massive carp population in that river is knocking them, the like natural recovery of them, back even harder. Because we'll go there during the season and sometimes in June, we'll see a few Vallisneria poking up in places that I didn't plant them. But then by the time we do our full vegetation survey in August, September, they'll be gone. And they're not just like randomly dying, they're definitely getting eaten. So there aren't too many, there's like no that I have seen persistent Vallisneria beds right now, again, other than the ones that we're actively planting, but that's why we're actively planting them. It's also not too many years post treatment. And some of these plants take a little bit of time to recover. But we're definitely seeing, I think, kind of a multi-pronged situation of some climatic differences, some reservoir changes. Again, we're pretty heavily impacted by how they release the water from the Croton res, and that has changed. That changed while we were doing the treatment as well. So it's hard to say. I'm sure the treatment knocked them back to the point where it's hard for them to recover now. But yeah, there are currently no beds. But we're hoping that given a little bit more time after the treatment, and we're really putting in a lot of material in there every year, that we'll get some of that recovery.
Okay, so we have a question for Lindsay as well. Was there any effect of DO early in the growing season?
I can't really answer that. Like, what's the word I'm looking for? Not honestly, but the DO measurements we took were very discreet. I just wouldn't really feel like it was like honest to say that there was any impact of anything. It would have been nice for us to use, like a long-term sort of like logger or something throughout the season before and during. But unfortunately, we did not have access to that. But maybe in a new plot, somewhere where there aren't plants, we could look at that. But we really just took like one measurement when we went and checked the plant. So I really can't say that there was any impact.
Okay.
Or effect.
Thank you.
Right. Next question is there currently a permit process in New York for native aquatic plantings?
So there's two things. it depends on where you're working. So for the canal, if you're working in the canal, any canal in New York State, then yes, you would need a permit for that. That would just be like a canal permit. But from, and then also wetlands, there's probably something with wetlands, but that's going to depend on how much you're doing. And so it would be up to your individual DEC region to determine if they thought you would need a permit for that. As far as for me, right now, I just have the canals permit. So it's really going to vary between regions, but there's no like designated as far as I'm aware.
Right. Thank you. So that's it for the questions in the chat. Do other folks have questions? Any other burning information? It's just need to find out. Okay, then I will go back to sharing so I can finish up the, let's see if I can get this to work out. Okay, hopefully that'll work. You can see my screen. I can make this go forward. So thank you everybody for joining us today for this webinar, and many thanks to Bethany, Ashley, and Lindsay for really informative and cool presentations. I'm very excited about all of this information and hoping this will be used by us and many other folks for future management decisions. So I'd ask that folks potentially stay tuned for session 2 of the autumn webinar series on October 28th, which is just next week already, which is going to focus on remotely piloted systems for aquatic weed management by Andrew Howell at North Carolina State University, and CEUs will also be offered. And note that the recording of today's webinar will be posted at the www.neapms.org webpage or website, sorry. And I'm also going to put in a plug for potentially folks joining us at our annual NEAPMS meeting at the Mystic Marriott Hotel and Spa in Groton, Connecticut on January 6th to 8th. The agenda and registration details can be found at our website. And we will let folks know that we are offering travel scholarships to students. So thanks everybody. And look forward to seeing you potentially next week. And don't forget to sign up for your CEUs. We have a lot of links in the chat box, so make sure you will go ahead and link on to that Teams location where you can sign out to get your CEUs.
Thanks, Kathy. I just dropped the link in the chat if anyone needs that.
Yay. Thanks, everybody. Take care.
Thank you.
Thanks.
Thanks.